Father, thank you for today and for everybody who's here. And Lord, we pray that you'd be with those who are not, if they're sick, for your glory and for their good. Lord, I pray that you'd make it interesting, that you'd captivate our minds and our hearts with just the wondrous things that you've done in creation. That you'd be with us and bless us, we pray in Jesus' name. Amen. All right, well. Okay. Let's see, we were on angiosperms. Last time we learned about all these parts, right? Yeah. Let's do a little review of their life cycle, okay? Alright. Tell me very quickly, since you're going to have to know it anyways. Stigma, this, style, this, ovaries. What's in here? Eggs that will become seeds. After the egg gets fertilized by sperm, pollen, which is made here. Just called, this whole thing is called a stamen. This is the anther. This is the filament. This whole thing, this female thing, is called a thistle. You got to memorize that. You just got to know it. These are petals. What are they there for?
Attraction.
Attract pollinators. Good. This is called a sepal. What's it there for? Protect the developing flower. Which part of this makes the fruit? What becomes the fruit? Which is the juicy part. And it's all is said and done. Stigma, style, ovaries, anther, filament, sepals, or petals. Which one? Fruit is an engorged ovary. Flowers make fruits, fruit makes seeds. Seeds make plants, right? So, flowers make fruit, fruit makes seeds. Where do you find the seeds? That's where you'll find the fruit. The seeds are made right here in the ovaries. Okay? Alright? Gotta know it. No time like the present to start learning it. Alright. Well, just a little more detail on some of this. If we're focusing on our anther here, then inside you've got this. This is all part of the sporophyte. This area right here is called our microsporangium. You know what? Forget this. I'm going to make you've got to know the one for the conifers, okay? We did that. We drew all that, right? You must know it. You must be able to label it. You must be able to tell me what's going on. With the whole mitosis, meiosis, it's not that complicated. If you practice. If you don't, it's a nightmare, okay? I'm not going to make you know this one. You do absolutely have to know the parts of this file, okay? What's the function of this? Let's start very, very big. What's the function of that? Yes. Yes. Okay? This whole thing is called a stamen, right? This part here is going. Here's really kind of freezing. Anyway, this part here is going to ultimately make the thing that makes sperm, right? Inside the pollen, this is going to make little pollen granules, okay? All over the place. When you go up to a flower and you get too close or you touch it or whatever and that yellow stuff gets on you, it's coming from this. And it's getting pollen on you. Inside of those pollen grains is where the sperm gets made. And so the pollen grain will ultimately come and will land here, and the sperm will release and they'll make their way down, okay? Everything about this is going on with the male, right? Okay? How much of the plant's DNA is in here? 1 in, 2 in, 3 in, 4 in? 1 in, right? 1/2 of its total amount of DNA. Okay? You look here. Got a little deal here. This is the megaspore mother cell. I don't even care that you know to call it that. What you need to know is right at that location is where an egg is going to be made. And there are other names, oogonium and all this kind of stuff. You just need to know this is where eggs are made. Again, an egg, 1 in or 2 in? 1 in, right? So sperm from a pollen grain, pollen granule lands here, sperm is released, begins to digest enzymatically its way down, finds its way here, it combines with this, and now we're going to begin producing. First of all, you got a zygote, right? But then you're going to make a seed. Okay, and that seed is going to be 1 in or 2 in? 2 in. Okay. We've talked before about the idea of it. You got the right idea of this. Of a reduced gametophyte generation, right? With the ferns, or excuse me, with the mosses, the gametophyte was the big part. With the ferns, the gametophyte was the small part. Here, you never see the gametophyte. You never with your eyes. It's contained inside of a pollen granule. It's contained inside of the ovaries. Never exposed to the open world. Completely contained. Okay, so dominant gametophyte generation. It's not even close. But very, very successful, right? You got to know this is literally sticky, right? So the pollen grains will actually stick to it. They digest their way in. This thing is going to make the seeds. It's going to then gorge and produce a fruit. We talked about how fruit doesn't just have to be what you think of. It doesn't have to be a juicy apple. It can be a stickerburr. It can be a pecan, a walnut. They come in many different types, shapes. There's simple fruits, compound fruits, multiple fruits. I don't even care that you know that, but since I threw those words at you, I'll tell you, okay? So a simple fruit would be something like a cherry. 1 fruit, 1 seed. 1 ovary, 1 seed. Okay? There's, or 1 fruit with multiple. There's multiple fruits, where out of 1 flower you're going to get a bunch of different fruits. You think of something like a blackberry or a dewberry or a raspberry. And then there's something like a compound where you've got multiple fruits, but they've kind of fused together, and that kind of happens like with a pineapple and stuff. So there are different types, there are different strategies, but it's really all about just protecting the next, because this is, inside that seed is the next sporophyte. It's the next generation. We've got to protect it, we've got to disperse it, we've got to distribute it, we've got to fertilize it. You can fertilize it just by literally the rotting fruit. You can fertilize it by the poop of whatever ate that thing. I'm sure I told you before, there are certain berries that unless they are, the seed coat is actually digested, it's subjected to hydrochloric acid in the stomach, it will never germinate. It must be eaten, which ensures that, that when it's eaten, it spreads somewhere else. Something like an apple tree actually must be subjected to freezing temperatures. It will not germinate unless it's in the right kind of environment. Okay? Again, you think of sticker burrs or things like that that will stick to fur or clothing, guarantees that it's going to get transported somewhere else. Just God's given amazing diversity to life and to ensure that it goes on. All right. Seeds. Do they have you know seeds? I would have you know seeds. Apparently they don't care about seeds. That surprises me. Seeds are cool. You don't think seeds are cool? Seeds are cool. Let's see what I can remember about seeds. Just off the top of my head. So you'll have, yeah, we need to know this. It ties into other things. So basically what we have right here is a lima bean. If you open up any lima bean, it looks just like this. Very, very convenient, okay? Most of what you see here is what's called endosperm. We need to do this too. Okay, so you're gonna have a seed coat. Seed coat's going to protect that thing, right? It's got to get wet, or it's got to get digested, it's got to get something. Something has to happen, because if I just lay a seed out there on dry ground, I don't want it germinating, right? It needs water, it needs soil, it needs all these different things, and so it needs to somehow have some built-in mechanism to respond to the environment. And so it comes down to water. If they're exposed to water, the seed coat will relax and water will get in, and then we'll have some activation of its enzymes and whatnot. Okay? All right. This little part here is called a radicle. And a radicle, all it is an embryonic root. So this thing's whole purpose is to find soil. Okay? If you plant a seed upside down, so that the radicle is facing up, and so that the parts down here with the little embryonic leaves are facing down. Well, that ain't right? You don't want your roots going up into the air and your leaves going down on the ground, right? So it's got to be able to adjust. So what we need to do, if we plant, and you don't know which side is up, which side is down, do you? You ever planted a seed? Usually you're just dropping a seed, right? Okay. So how does the plant make an adjustment? This part has to quickly turn, doesn't it? And head for, as it grows, it's got to find soil. And this part can't bury itself in the soil. It's got to find light, right? Okay, so it's got to grow up. And then pretty quickly it needs to form little leaves, right? And then other things need to come off. That has to happen quick. And it can do that. You don't have to come in and turn the plant right side up after it germinates. It's really cool. This is called gravitropism. So tropism refers to movement. So it's moving. What does gravity sound like? Gravity. So it moves towards gravity. It's that simple, right? So this thing's going towards gravity. And this is doing something called phototropism or heliotropism. What does photo sound like, or helio?
Photo light.
Towards the light, or towards the sun. So this part is going to seek out sunlight and grow towards it. Erase that, but moving toward light. Moving toward light. Okay. Okay. So I'll let you finish that. So we've got our radical, our embryonic root. It's got to find soil. It does that through gravitropism. It literally senses gravity and grows towards it. Up here, we didn't label this part. It's called a cotyledon. Cotyledon. And a cotyledon is an embryonic leaf. And the other part is our epicotyl. I guess I could just do this. Epicotyl, and that's an embryonic stem. That only leaves one other thing. So all this so far that we've been talking, we talked about the seed coat, we talked about these guys. By the way, it wouldn't actually be green, okay? It's going to be white. It'll become green once it shoots up. In fact, my son did a science experiment on this last year, growing. He had to grow some plants, and he had to grow one like in the refrigerator, and he had to grow one in a cabinet, and he had to grow one in the sunshine. And what was ironic is the one in the cabinet grew the largest, the fastest, because it was desperately seeking out light and finding none. So it used all of the energy that was stored in that seed to try to find the light. Never found it, it died. Guess what color it was? The whole plant. It was about this long. It was pure white. The one in the ice box grew only a little tiny bit because it was so cold. Your enzyme, by the way, I talked to my friend and we did talk. Yeah, got lots of information. So the one in the ice box grew a little bit, it grew very slowly. The one in the sun did not grow as fast. It quickly found light and became green. One used all of its energy just trying to find the sun. The other, once it found the sun, used its energy to start making chlorophyll, which makes it green so that it could do photosynthesis so that it could survive, okay? So, cotyledon, embryonic leaf, hypocotyl is just the stem below it. The radical, it doesn't even seem fair that you would need to know these names, right? I don't even know that I care if you know these names. I may give you the names, but I want you to at least tell me what they do, okay? This is an embryonic leaf, this is an embryonic stem, this is an embryonic root, right? I do want you to know gravitropism, and I do want you to know phototropism. Okay, these are adaptations that God has given to these plants. The root, literally, if you plant a seed upside down, the root will find gravity. And this will shoot for light. And if you turn it, if after it found it really quickly, you buried it again really fast, it would flip again. Like it will find it, it will follow gravity. And they've done experiments like in space. You're leaving us?
I have an appointment today. This is the day.
Okay. So we think of the song, "This is the day that the Lord has made." It's from Psalm 118. All right, anyway, I can just go on and start teaching you guys about the Psalms now. Okay, so cotyledon, hypocotyl, radical, seed coat. Last thing on this that you need to know is all this stuff in the background. Basically, the biggest bulk part of the seed is, I'm coloring it blue. It's not blue at all. It's just white, right? But it is called the endosperm. Endosperm. And it is the food source. So how can a seed live for years without dying? Well, it doesn't need to do a lot of metabolism, and it has a food source. How does this thing that's white
and can't do photosynthesis at the first, how can it grow towards the gravity, and how can it grow towards the sun? It has a food source. It's not unlimited, right? Remember I told you, the boy when he did it, he had the seed, he put it in the icebox, he put it in the light, he put it in the cabinet. The one in the cabinet grew huge, trying to find light, until it ran out of food. And then it died. It's that simple, right? It has a certain amount of food. It's certainly enough to get it going. This would be analogous to a yolk sac in an egg, okay? That yellow part, that is the food. That chicken's just stuck. We just got chickens the other day. Yesterday. Totally excited. One of these days we'll raise our own eggs even, like, you know, fertilized eggs. Well, that chicken, that zygote of the fertilized chicken is like tiny in this egg, and it can't eat. You can't put the chicken feed in there. What's it eating on? It's eating on yolk, right? If it doesn't have enough yolk, it won't develop. When it runs out of yolk, it better hatch out, right? Okay. So, yeah, that leads us to the next thing. Let's talk about these angiosperms. Can't believe your book didn't talk about all this. You must know this. You don't need to know the names, but you need to recognize the names and tell me what the things do, right? Did you make yourself a note of that? I'm promising you that for a test. You'll see these things on the test. I won't make you memorize hypocotyl, cotyledon, radical, but I'll label it for you. You've got to tell me what it does. Okay? Which one of these grows towards gravity? Which, you know, I don't know, whatever. We'll see how I label it, but you get the idea. Okay. All of your seed plants can be divided into gymnosperms and angiosperms, naked seeds and covered seeds. All of your angiosperms can be broken down into two groups, monocots and dicots. Okay? So we'll go back up to our big thing here. We got plants, right? We've got non-vascular. We've got vascular. Undervascular. We have seedless. These are the ferns and stuff, right? And then we have seeds. And then under seeds we have gymnosperms. And we have angiosperms. Your gymnosperms, you got stuff like Coniferophyta. Mono means what?
So,
No, that's auto. You're in the ballpark.
like one.
One, yes. So this is monocotyledon. And this is dicotyledon. What does di mean? Two. So one cotyledon, two cotyledon. Well, we've seen this word before, right? Cotyledon. It's an embryonic leaf. Does it have one embryonic leaf or does it have two embryonic leaves? All of these can be divided into these two groups, and they have certain characteristics to them. So there's a reason that I want to go to the seeds. You can look at a seed and know a whole lot about what that plant's going to be like. Good question. She gave good advice. Not all the answers, really good advice about how to proceed. I just thank the Lord because I was sitting there going, I don't know anyone even who could help you. And I was like, oh yeah, you do. You have a friend who did this. Okay. So we're going to go here. We're going to go monocots. We're going to go dicots. Okay, so so the number one characteristic is that this has one embryonic leaf, right? This has 2 embryonic leaves. Some other things that are associated with this. These guys will have fibrous roots. These guys will have tap roots. These guys will have sort of want to use. Just talk about it. Let's see. Where's it? Just skip over this. Oh, it does talk about monocots and dicots. Good. Okay. Yeah. Okay. This one has parallel veins. And this one has branched veins. This one has scattered vascular bundles. This will have a ring of vascular bundles. This one will have flowers, flower petals, in multiples of 3 or multiples of 4. This one will have flower petals in multiples of 5. So let's draw these in a second, what each of these would look like. So right now it's just a bunch of words, right? Start at the root level. A monocot is going to have roots that just do this. Whereas a dicot is going to have a root system that looks like this. What does that look like? That you eat? Carrot. A carrot's a dicot. A carrot is a very definition of a taproot. Okay? A taproot means it's driving deep. This lets it get a good, firm anchor. Okay? And then it can spread out from there. But that's the very essence of a taproot. This is a fibrous root system. It's got a lot of little tiny tendrils that are sticking down. Okay? Now, I say tiny. These can actually be huge. These can go potentially over 1 meter down if it's alive long enough, right? All right, so we've got a taproot and we've got a fibrous root. Next thing, we want to think about, just had it, yeah, there it is. You're going to have some kind of a stem sticking up above ground, right? And then you're going to have some leaves. Well, if I look at the leaves on this thing, the veins that are running through it are going to be netted. Whereas over here, any leaves, the veins, are going to be running in parallel. What does that look like so far? Especially if it's a little short to the ground. Let me redraw it. Here's the ground, by the way. What does this look like? If it were green, about 3 inches long, you hit little white balls with clubs all around it, or you stood on it at the Wimbledon and you hit balls off of it. It's grass. Grass is a monocot. Grass has all these characteristics. It's got these little fibrous roots. It's got parallel veins. You can see them. If you pluck a piece of grass, you hold it up to the light. Let's go outside. Come on. This is better if it's real. Let's go. Come on. So much for this recording. Come on, we gotta go quick.
Here's the drawing.
So if we were to do a cross section of a stem, okay? So this part here, and we're going to do a cross section. What you'd find is vascular bundles arranged in rings. Okay. And here, if we were to do it, you'd find them kind of scattered randomly throughout. These are essentially the veins, the xylem, the phloem flowing through, transporting stuff. In dicots, it's always in a ring, and monocots, it's randomly scattered. Okay. So, you know, something that I would be fairly typical of me, would I'd give you enough information, I'd give you 2 or 3 facts about a plant, or I might show you a picture of a plant, and I go, well, what is it? Monocot or dicot, right? If I tell you that it had 10 petals, you tell me it's a dicot. If I tell you it has a taproot, you tell me it's a dicot. If I tell you that it has its veins scattered randomly in its stem, it's a monocot. Okay, I might have said it wrong, sorry. If I tell you that it has, you know, fibrous roots and parallel veins in its leaves, it's a monocot. I tell you it has 9 leaves or 9 petals, you tell me it's a monocot. Okay, all right. All right, is there anything else your book thinks you ought to know? Nope. Okay. Y'all ready? Okay, time to, we got a half hour to do an intro to animals. Let's do it. Turn the page, draw a line, whatever. We're done with plants. We're moving on. Okay. We haven't talked a lot about this yet, but we need to. Organelles are subcellular structures that are highly specialized. They're made out of, if you all haven't written that down or aren't writing it down, you make a mistake. You must have this down. You must tell me this in order. Organelles are subcellular structures, right? Made out of proteins and nucleic acids and hydrocarbons and sugars. They're things like ribosomes. Nucleus, cell membrane. They're little parts. They're like organs for a cell. All cells have organelles. Eukaryotic cells have membrane-bound organelles. Prokaryotic cells do not. That says complexity. Probably doesn't look like complexity. I probably ought to fix that. And specificity. Complexity and specificity. So organelles make up cells. For some organisms, that's the end of the road. A cell is the organism. This is true for bacteria, it's true for protists, right? I can take the most complex of protist life. I can take something like a kelp, and I can pull it apart just one cell, and that one cell is going to be just fine. That one cell can do absolutely everything that the whole organism can do, or the whole colony of organisms can do. It's not truly multicellular. It's just living cooperatively. Right? It'd be kind of like you if in a household, right? Maybe one person cooks, one person does laundry, one person builds things, one person burns things. Yeah. But if you had to, you could eat a grilled cheese sandwich, right? Boil some water and make whatever. You could do what you needed to do and you'd be fine. That's very true for protists. That's not true for you. There's no cell in your body right now that I can just go pluck and it can do everything and continue to exist. Your cells are highly specific, specialized, okay? You have layers of complexity to you, as do all animals. So organelles make up cells, cells make up tissues. Tissues are collections of cells where all of the cells in that tissue are specified to do a certain function, right? Name some tissues that you have. Do you know of any? At all? Muscles. Connective tissue. Your integumentary system with the layers of tissue in your skin. Your nervous tissue that's conducting impulses. Think about just what it takes for a muscle to move, okay? For a muscle to contract. Let's answer this question. What tissues are involved in muscle contraction? Okay, well what do you have yet? What's the baseline? What must you have for a muscle to contract?
Muscle.
You must have muscular tissue. Okay? What do your muscle tissues need to have in order to be able to contract?
The ones who need to move,
Pardon?
the tissues who need to move,
Well, that is the muscle.
yeah. The ligaments, is the tissue.
Well, to make the bone move, you need that. But for the muscle itself to just go, you know, like when you get a cramp, or if you ever have a muscle that just starts pulsating for like no good reason, right? Okay. What does that muscle have to have? Huh? Nerves. It's got to have something to send the signal, right? So we've got to have nervous tissue that's going to tell it to contract. But you know what? Oh goodness. Once saw an alligator killed. Okay? And hours after this thing was dead. And the backstrap was put on ice. The thing seized up, tensed up. The muscle still contracted, right? That tissue, even though the animal, the organism was already dead, that tissue was still alive. And it was able to respond to stimulus from the nervous tissue, which was able to respond to stimulus from temperature, which is wild to think about, isn't it?
Mhm.
But in 5 hours, that's not going to happen anymore. What changes over 5 hours? I still have muscular tissue, I still have nervous tissue, but there's some other factor that after a certain period of time, I can't just juice it, right? If somebody's dead, and they're dead for, just a few moments or whatever, you can come with the paddles and you can charge, and you can, even if you're not able to revive them and bring them back to life, you can still make their muscles twitch. But after they've been sitting in the morgue for a day or two, I can't do that. What's missing? The blood, which is bringing the nutrients, the metabolism, the energy, right? Blood is part of your connective tissue, right? So that's 3 totally different, highly specialized tissue types just to contract that muscle. Now the muscle is one tissue type. Connective is another. Nervous is another. They're coming together to form an organ in something like your heart, right? You've got muscle, you've got blood, you've got nervous tissue. I mean, it's just doing what it does. And if it ever goes wrong, you'll know. But you're young, you're healthy, it's just sitting here doing its job all day, every day. You don't think anything about it. Look how far we've already traveled down this, right? And then you think about organ systems. So you've got organs that are interconnected. You think about your muscles throughout your entire body. You think about your skeletons. You think about your skin. You think about your cardiovascular system. You've got a heart that's doing things with lungs. It's doing things, and then you've got like kidneys that are filtering stuff out of the blood. And kidneys are taking things down your ureters and into your bladder and into your urethra. And then, and you're wildly complex. But you're not alone. Nothing that we've talked about so far is present in you that's not present in a crayfish or a fish. In fact, other than specific organ systems, it's the same in a plant. A plant has organ systems. A fungus has organ systems. They're not as complex as you, right? They're simple. But as we move down here, we increase in complexity and specificity of cells. One cell cannot do everything that it takes for you to be alive because you are so complex. Bacteria are so simple, so they can. Simple is a good model, but this is, look at you. Look how cool you are. So, all right. Organelles, cells, tissues, organs, organ systems, organisms. You need to be able to tell me that in the right order. Hopefully you understand why this is important. Where do they want you to know? All right. Okay. All animals, right? All. You know what this is, right? Egg plus sperm equals zygote. Question is, what happens from here? Okay. So the zygote, the cells will begin to divide without growing. Notice that we always draw the egg huge and the sperm tiny. One thing we didn't talk about even is the process of ovulation. We've kind of just skipped this. but there was this idea that you've seen, at least brought up before, of a megaspore. This is in plants, okay? Megaspore mother cell. Or if we want to think about it in humans, okay? You've got, you know, here in your ovary you have you got a cell, or it could be for a sperm. It goes through meiosis. Meiosis, you're going through 2 rounds of cell division. This is not merely cytokinesis. Okay, you go through 2 rounds. So after round 1, typically you'd expect, well, I've got a cell like this and this, and then after round 2, I've got a cell like this and this. I get 4 equal cells. That's not how it works. It does in sperm. In sperm, I'll have one like this and one like this, and then I'll have come down here and I'll go, okay, well, I got, well, I guess it would be like this, wouldn't it? It'd be divided into 4. I'll take 1 cell and I'll get 4 equal sperm cells, and they'll all be 1/4 the size of the original cell. It's not how it works with eggs. With eggs, so this is wrong, okay? With eggs, how it works is this. After 1 cell division, I'll have 1 huge cell, I'll have 1 little teeny tiny cell that's going to die. And after 2 rounds, I'll have 1 big cell, and I'll have a little teeny tiny cell, and then these other 2 little teeny tiny cells. 3 of these cells are not going to make it because I need everything in one. I need all my basket in 1 egg. This has to have everything it takes for this zygote to grow, or at least to be able to divide and to function without growth, without doing a bunch of metabolism and stuff, without getting food, because it's going to go through rapid cell division. Rapid cell division as soon as it's fertilized. So I take my big old egg and my itty bitty sperm. All this guy is really contributing is genetic information. He's not bringing any cytoplasm, he's not bringing any organelles, he's not bringing anything to help the zygote survive, just the DNA. Okay? So mom gives half, dad gives half, here's the baby, the zygote. It's going to rapidly begin dividing. 1 cell is going to become 2, 2 is going to become 4, 4 is going to become 8, right? And then 16, it's exponential growth. It's going to form something called a blastula.